Welcome everyone to another Systems Neuroscience and Complexity talk. It's my great pleasure today to have Sid Joshi joining us. I remember a couple of years ago when we were trying to think about the fluctuations in pupil diameter and their kind of systems level impact of noradrenaline and trying to sort of link together those circuits. There were really a few papers that stood out to us. There's a McCormick and McGinley review and then some empirical work in neuron that was really influential, some work by Jake Reamer, and then Sid's paper with Josh Gold, where they had these really beautiful recordings that were linking pupil diameter to firing in the locus coeruleus, but also all of these other areas like the superior colliculus, the inferior colliculus, the anterior cingulate, that I think really expanded the way we were thinking about it. And rather than trying to focus in on one individual area as this kind of precise readout of the pupil, or maybe even controlling the pupil diameter, embracing the kind of biological complexity of the circuit and thinking about the sort of more distributed patterns that we could maybe read out or infer from pupil diameter. So Sid's paper was just so influential for us in thinking about that. And then just recently we had some fun chats with Jake Reamer and Matt McGinley and co, and Sid was there, and so I took the opportunity to try to get him to come and chat with us about some of his ongoing work. So I'm really happy that you could join us, Sid. Looking forward to seeing what you've been up to and hearing more about your ideas in this space.
Thanks, Matt. Okay, so I'm going to talk about a bunch of measurements I've made in locus coeruleus and in cortex, while also measuring pupil size. And all of the work I'm going to show you is from the time I spent in Josh Gold's lab at UPenn. So I'll just start with some acknowledgements for the people there. So Josh Gold, of course, for being an incredible mentor and giving me kind of the space and time to really collect these data carefully without feeling rushed. I think that helped a lot in just making the measurements well. And Rishi, who some of you might have seen his name on a LC countermanding paper from 2014, who was one of his grad students actually, and had left the lab by the time I joined, but he had worked with Gary Aston Jones previously, recording from LC in monkeys, and he kind of really set the stage for others to come in, others like me to come in and record from LC. And it is a little bit of a dark art in the sense that you kind of have to know what to look for in these trajectories, which really, are far, LC is really far in. So EPIS, you're kind of doing recording in the dark, basically. And he had sort of figured out a really nice technique that includes making custom electrodes that allow us to get all the way down there repeatedly without causing a lot of damage. So that was Rishi, and then others, in the lab, and probably most importantly, the monkeys, without whom none of what we found out would have been possible. So shout out to Sprout, Sistra, and Oz. Some of them are still going strong, so and contributing to all of this. So the approach for these studies is multi-region recording and pupilometry. So the regions that are recorded from are either in the brainstem, one of these regions, pericolliculus, inferior colliculus, or locus coeruleus, and all of these are fairly deep in the brainstem, with LC being the furthest down. And so this is, of course, I should have said, a sagittal MRI image of one of our monkeys. And anterior cingulate cortex is, of course, further up in front. And if you look at these in and sort of an atlas view, so if you take a section through these regions, and then rotate it so you get a kernel view, that's kind of what I've shown you in these schematics here. So although this looks kind of large and bigger than the cortical map, it's actually just this little chunk of region here. And you can see it's really busy with a bunch of different little nuclei, all of which do incredibly important things. And LC is, of course, located all the way down here, just below the trigeminal tract. And for ACC, we picked essentially dorsal anterior cingulate cortex, which is a little bit dorsal to and then lateral to and below the cingulate sulcus. So I'll go into a little bit of how we record from these regions, but not a lot, because unless someone's super interested in some of the details of how we find our way there. Happy to talk about that. I'll briefly touch on it in a later slide. But first, briefly motivate why we picked ACC. I don't think I really need to sell it to you guys, but for me, two really important reasons. And one, I'll start with the second one first, actually. A lot of previous work in humans, in monkeys, and mice with different techniques has sort of narrowed down on identifying ACC as this region that encodes some really interesting factors that are related with goal-directed behavior. So, for example, different kinds of error signals, essentially in a performance monitoring kind of task. Sorry, not task, role, I should say, for ACC. And so that makes it super interesting to figure out, what are the neurons actually doing under different conditions. And also the fact that it's this kind of activity that's being recorded and reported from there is also similar to the kind of activity that's associated with locus coeruleus and pupil linked arousal. So the behaviors that are linked with LC linked arousal are also very similar, identical to those that are linked with ACC. So that's one. And the second one that I'm actually super interested in, and I think it's heavily underexplored, is this. We know a little bit, at least, not enough by any measure, about how ACC is connected with LC. And in fact, other cortical regions are connected with LC. So the canonical kind of picture with most neuromodulators, including norepinephrine from LC, is that it's sort of a one-to-many kind of broadcast signal. And I mean, that's also kind of getting refined a little with better measurements these days, but it's still broadly true, when there's sort of an autonomic effect, sort of a bottom-up, say, startle or surprise event or a noxious event. That really drives LC, and then that releases norepinephrine throughout the brain. It's sort of a wake-up signal to pretty much every area, cortical and subcortical. Now, in the reverse direction is where it gets really interesting, because from what we know, from tract tracing studies so far, it's mainly these prefrontal regions and cingulate that have descending projections to LC. In fact, primary sensory cortical regions don't have any inputs, or probably really weak, so they don't show up in simple tract tracing studies. And so that already sort of shows kind of a really interesting circuit, which is that if you have bottom-up regulation via LC of cortical regions, that's going to influence everything, sensory encoding, some more cognitive stuff happening up ahead. But in the reverse direction, if you think of what drives LC, there's a whole lot of subcortical regions that do that. But in addition, if you're looking at sort of the higher level processing in some sense, then it's really the prefrontal regions, medial prefrontal cortex, ventrolateral prefrontal, orbitofrontal, and anterior cingulate that have the strongest known projections to LC. So under some conditions, say of cognitive surprise or changes in performance monitoring during these more complex cognitive tasks, it's these regions, I think, that are going to drive LC. Whatever the autonomic tone, there's going to be an overlay of this cognitive descending signal that then drives LC kind of in a loop to then broadcast norepinephrine to the rest of cortex. So I think that thinking of it in that kind of loop has not informed, I should say, a lot of studies, especially the ones with recording and even simple behaviors in mice or in monkeys. And I think that's going to be a rich field to explore in the coming years. And as for pupilometry, I don't think I need to motivate it too much here. I'll just briefly point out that it has been used as kind of a readout of internal state for a very long time, including these very early attempts at quantifying changes in pupil size from Hess and Polt. And since then, it's been used really widely, not just for studying age-related or neuropsychiatric disorders, but also cognitive function, intelligence, memory, and so forth. So pupilometry is kind of important to understand in any of these contexts. So what we've done is to make these measurements mostly in kind of a baseline state. So all of the data I'm going to show you is with monkeys doing a pretty simple fixation task. It's essentially the same task we used in the data we showed in the 2016 paper. And so the trials are essentially fixation trials. A dot comes on in the center of the screen, and the monkey has to fixate for 3 to 5 seconds. And then on some randomly chosen trials in any given session, we play a startle stimulus. That's just a loud sound that turns on after the monkey's acquired stable fixation. So he's, it's after the saccade to fixate, and then about 1 to 2 seconds in, this sort of sound goes off. And we found, and of course others have shown for many years, that these kind of stimuli drive pupil dilations and also LC responses. So I'm going to show you these measurements and summarize the findings briefly, beginning with the data we've already published relating LC with pupil size. And if any of you, of course, have questions about that study or details, as Max said, feel free to interrupt and we can dive into some of those. And with the newer data, I'm going to show you that LC firing and LC-linked pupil changes are not linked with changes in single neuron firing in ACC. But if you look in a consistent way, there are changes in all directions, but there's no clear effect. But there are changes in coordinated activity in ACC that are linked with LC firing and with pupil size. And I'll briefly touch on some pilot data that I collected for a simple change point task with, so it's more behavior than just fixation. And in a set of experiments, I was able to do microstimulation in LC and show that that affects this behavior during a change point task. All of that was from one monkey, so we're not quite anywhere near publishing that, but it is really interesting that there was any effect, even with this very simple task. So these are some slides related to finding LC. And again, like I said, I'll breeze through them. If anyone's interested in these, I'm happy to come back and dive into the details. But briefly, there are signatures, both functional and also just purely electrophysiological, that kind of tell us where we are along a track that crosses these regions in the brainstem before leading to LC. And for example, the LFP has a characteristic profile in response to the startling sound in the 3 of the major regions that we encounter. So the superior and inferior colliculi, and of course LC. And those differences are the form and polarity of the LFP in response to startling sounds. So in SC, there's kind of a weak response, which you kind of expect because it's mainly oculomotor or visual. In Pericliculus, there's a clear auditory response because it's part of the auditory brainstem. And in fact, there's a biphasic response, not biphasic, I should say. There's an on and off response to the sound coming on and turning off. And LC has this kind of biphasic response, an initial dip, and then this large sort of hyperpolarization associated positivity. So these are really reliable kind of indicators of where we are in combination. Sorry,
can I ask it? I've been reading a lot about the superior colliculus a little bit just recently, and it seems like there's a lot of differences in different layers of the colliculus. So it's in the sort of superior part of the superior colliculus, there's a lot of connections with the thalamus, whereas much in the sort of deeper motor layers, there's more sort of descending projections to, the MLF and oculomotor structures and things of that nature. I wonder whether you looked at, because I see you're sort of crossing across a few of the different layers of the colliculus. If you could get to that level of resolution, or is it sort of tricky to? Is this sort of more like an LFP? So it's sort of broadly in the colliculus, but we don't quite know what layer we could resolve.
We can actually tell. So so this trajectory, which we've kind of settled on after Rishi's original experiments. If you look at the atlas, these regions look like they abut each other very closely. But in fact, when you have an electrode sort of going through these areas and you're listening to the activity, which is how I trained to do it, there's actually quite a bit of silence between the bottom of posterior cingulate, which is CG here, and the top of SC, superficial layers of superior colliculus, it's nearly a millimeter of silence. So I don't know if that's really, I think it's neuropil. It's not, sorry, it's not neuropil, it's space. And we're going through CSF because even if you're in white matter, you hear sort of activity in the background. You can tell it's still in the neuropil. And when you pop out of it and then pop into the top of SC, it's a really clear, amazingly clear signal because there's complete silence and suddenly you burst into superficial SC. And you can actually tell because you don't see saccade-related activity there at all. So if you just turn off all stimuli and the monkey is sort of searching around on the screen for what to do next, then you basically, if you take a flashlight and sort of turn it on in the room, you can hear responses to the light going on and off because superficial, I should say, SC has inputs from the retina and also descending inputs from layer 6 of visual cortex. So you're going to get visual responses there. And then as you go deeper, about 300 or 400 microns in, then you start hearing these zippy sounds as the monkey starts looking around. And it's the oculomotor bit. We aimed mainly for that. I kind of kicked myself for not recording in superficial SC. And yeah, anecdotally, I can say it's a little harder to get isolations there. You get a lot of good multi-unit activity, but you start seeing really nice, big, clear, isolated spikes only further down in intermediate and deep, which are the visual, motor, and motor layers, like you were saying, that have projections then down to the other oculomotor regions.
Yeah, it's super awesome to hear that. I got to sit in on a deep brain stimulation surgery back when I was working a bit more in Parkinson's disease, and we were trying to find the subthalamic nucleus. And I know exactly what you mean, where you're kind of like listening to the spikes and the kind of probes descending, and it's like spiking like crazy, and it goes silent, and then it starts to spike again, but in a different way. And it's really fun to kind of think about that as like a kind of, the practical challenges of trying to like advance this little electrode kind of blindly in a way. You're almost sort of like using sonar or something to try to figure out where you are. It's kind of cool. So just as a follow-up question on the colliculus, Doug Munoz has a kind of an interesting kind of research program where they're thinking about the colliculus and inhibitory connections to the Edinger-Westphal promoting pupil dilation. And are they a part of the motor colliculus? Do you know kind of specifically about the kind of layers that are implicated in that? I've never really kind of had the time to really dive into that. I was just curious how this might all fit in together with the pupil story.
I think in his data at least, so the work with I think it's Wang and Munoz, there's a series of papers. Those are from stimulating intermediate layers as well. So it's the same area that we recorded in for the 2016 paper. And in fact, he and I have chatted about this a bit because I think in the initial series of papers, they had no recordings in there. They did microstimulation or behavior, and they tried these sort of salience stimuli where they could modulate the salience of the stimulus for the animal, and they find that affects pupil size.
So that's cool. Awesome. All right, I'll stop interrupting. That's great. Thanks.
More to gossip about it later, but that's some other point.
Sorry. Yeah. So I have a question as well, kind of on this slide, but I feel a bit bad because we've kind of stuck you here. It's similar to Mac, but more related to the locus coeruleus. Is that okay?
Oh, totally. It's all good. Mm-hmm.
What I was wondering is when you're sort of figuring out, when you're in the LC or not, do you ever differentiate whether you're sort of or see a difference between when you're in the LC proper versus the coeruleus region? So the reason that I'm kind of asking that is that I've wondered about it. I don't know the organization in monkeys or rodents, but in humans, from looking at sort of post-mortem data, you see this kind of, at the sort of caudal, so the bottom end of the human LC, you see this sort of, fanning out of cells that I guess is this sort of coeruleus region, right? And it's not defined by a really sort of tough anatomical boundary. And, it's sort of full of all these, LC and noradrenergic sort of cells and distal projections from the LC. And I've read from, you know, in the animal work that you do see this kind of spontaneous firing and stuff in that region. So it would be very LC-like, but I understand it also has quite distinct, you know, afferents from different brain areas and things like that. So I guess, yeah, my question is, do you differentiate between that, or do you ever sort of end up there and, you know, realize that you're not in the LC proper?
Great question. I wish we could differentiate, because really with the monkey work, it's a lot. There are techniques. So the short answer, there are techniques to do it. We haven't done that yet. What I can say is I suspect that there is going to be a difference between sort of the core LC and the peri LC region, especially given this really nice work from Regan Kasur's lab at MIT, where they recorded these GABAergic cells in the peri LC region, and they actually show that they're functionally, in some ways, functionally distinct when related to pupil as well in mice. In my data, the one thing I did notice that was interesting, but I haven't had time to dig it out. I probably should, because that might be getting towards an answer for your question, is that in almost exactly the same trajectories on different days, I find at the level of LC and with response properties that are very LC-like, neurons that have different spike waveforms. So there's this canonical picture of LC neurons having broad waveforms. But I found LC neurons that have narrow waveforms, but exactly the same kind of firing rates and responses to startle stimuli. But I wonder if those are, you know, the kind of parvalbumin, GABAergic, not noradrenergic neurons in the, I guess I'm trying to remember, they're more medial to LC, I think, this cluster of GABAergic cells. They might be the ones that, you know, we've encountered with that have different waveforms. And I wouldn't be surprised that the response properties are different, because I expect that entire cluster of neurons, you know, the core and the periphery get similar inputs, similar-ish. So they're driven in maybe the same way. But what would be interesting is where they're projecting to, or is the GABA, you know, some people call it the shell, I think. Is that basically just regulating LC, you know, preventing it from running away and making people anxious all the time, something like that. So yeah, functionally, I'm not entirely sure, but I have, you know, I suspect some of the cells we have in here and all of this data are not core LC cells, actually. Just
Thank you.
based on
That was a really awesome answer. Thank you.
So this is a little bit more about the functional mapping on the way down. It's again related to you know, identifying the regions along the track and making sure we know where we are. So this is what Mac was asking about. So we've recorded mainly in the intermediate or deep layers. So this is an example from the deeper layers of SC. So what I've plotted here is these are PSTHs, so just average firing rate of single neurons in superior colliculus, actually of one single neuron in this case. And the 8 plots are for locations of the targets of visually guided saccades. So here the monkey fixates and then has to saccade to one of 8 targets in any given trial. And what you can see is that there's this pre-saccadic, classic sort of superior colliculus pre-saccadic burst for these 2 locations, but not for the others. And that's because SC has this really beautiful map of saccade endpoints and amplitudes that was discovered in the 1970s sometime by David Robinson. And it's incredible. Every time you find SC, you know exactly where you are based on these maps. And if you have the monkey fixate and then do electrical microstimulation at that spot, you can evoke saccades to that same location that the neuron encodes. So that's SC. And I do have videos of these, but I don't know how much it'll add to the time in this talk. So I'm happy to show some of them if you guys are interested, but we can also come back later if you want to get moving. Any preferences?
It's totally up to you, man. Whatever you'd like to do. It's we're all sort of lots of different backgrounds here, so I think it's always cool to see real data. But if you want to kind of get to the implications and have a chat about that, that's also cool with us. So choose your own adventure.
Okay, will do. So I'll show some more. Well, these are essentially like mapping or functional characterization again. So after SC, there's Pericliculus, which is of course auditory, and here I've plotted these are raster plots aligned to the time of a broadband burst of sound. So what you can see is when the sound goes off, that gets a strong response from this site in Pericliculus. And the other things plotted here, and this is the LFP, this is the PSTH, so it's just the average of all of these trials, and pupil size. And this is another sort of canonical response type that's seen in Pericliculus. It's where the spontaneous activity of a neuron is suppressed by sound. So these are cells that are inhibited by sound in Pericliculus. Then moving on down the track, there's the trigeminal tract, that's ME5. So it's the tract that carries the fifth cranial nerve. And this is the nerve that innervates the orofacial muscles. So it's kind of really cool to observe the monkey when the electrode's in here. So it's a motor nerve, so it's very active. It's just buzzy all the time. And then if you give the monkey a sip of water or juice reward, and he starts sipping at it, you can hear the neurons modulating to that. So you can watch the jaws move, and you can hear the neurons responding. And if you microstimulate there, you actually see that elicits a sipping kind of jawing action. So you can be pretty certain where you are in the brainstem if you cover sort of all of these sort of mapping steps, I guess, in a way. And if you stray kind of a little bit medial or lateral, it's not the right track, because if you go medial, you get into the trochlear decussation. So that's the fourth cranial. That's the trochlear nerve, rather, that innervates the extraocular muscles that drive adduction and intorsion. So if you record there, you get responses to eye movements that are down and left or right, but nothing to up and left or right. So they're really remarkable signatures of the function of these regions, and it's actually a lot of fun to do these recordings for some anyway. I'm not sure it interests everyone, but and now I'll get to LC. So I'll show you a video here of microstimulating LC and eliciting pupil dilation. So these are data that we showed in the 2016 paper, although not the video. Let me pull this up. Oops, it's on the wrong screen. Just a second. All right, can you all see that? All right, so what you're seeing here is this is, if you're familiar, you might be familiar with the Ilink tracking system. So this is just an image of the pupil on the monitor that's connected to the computer that runs Ilink. And the image is pupil stabilized. So it's a little weird. You'll see the background kind of moving around and the pupil stays stable. And this LED here is, it's going to go off every time there's a stim pulse that's delivered through the electrode. So the electrodes in LC, we've already recorded activity there. And now what we're doing is having the monkey fixate and apply a tiny bit of electric current through the electrode. And so every time that current's delivered, you'll see this LED go off. So I want you to look at the pupil, but sort of attend to the dot and see what you see. So you all see a little bit of a loop every time the stim pulse is delivered. Okay, I will. shall I run that again? Did everyone catch some of the dilations? All good?
It was quite interesting noticing like, there's like all these like subtle, tiny micro changes over time. And then you see this like little sort of swelling and then, but it quickly dilates again afterwards. I think, I mean, is that kind of characteristic to what,
So you
what you normally see?
the kind of pupil oscillating a little bit all the time anyway, and then changing? Yes. In fact, I'm going to come to that again in a bit.
And then I guess as well, another thing that I noticed is that the pupil was quite dilated to begin with.
Yeah.
And if the animal was, let's say, in a much brighter environment, would you expect that the evoked dilation would be of a larger amplitude? Like, do you see like something that Gabriel, a grad student in my lab, and I talk about a bit is this kind of idea that as the pupil becomes more dilated at rest, there's almost sort of like less capacity for it to dilate with a phasic burst that might be proportional to its kind of starting point, so that if it's really constricted and you hit it with LC, it can, noradrenaline can like expand, but if it's already pretty dilated, it only has like a little bit of room to move. And I from vague memory, there was something like back in the 90s that Gary S. and Jones or someone like that did that kind of argued against that kind of naive interpretation that there was more subtlety to it. But I'd be curious to kind of hear your take on what you've seen.
So there's a lot there actually, and that's super interesting. So there's in the 2016 paper, we did one analysis. It's I think in the very first figure or the second figure, where we look at spontaneous oscillations in the pupil, which you were pointing out just a minute back. And in fact, the spontaneous dilations are larger when the baseline preceding that dilation is smaller. So it's definitely, I don't know what Gary's view on this is because I haven't talked to him about it, but it's definitely true in the data that when you have a smaller baseline, you're going to get a larger, both spontaneous and evoked dilation. Now, I really wish if we'd known a little more about this when we did those experiments, we would have done something parametric, like changed in blocks, had the background luminance different or graded, say, in 5 steps to set the baseline pupil size at different starting sizes. And that's something I'd still want to do at some point. We haven't done it yet. So in all of these data, the background was basically, was actually dark, black essentially. And the fixation dot was a white spot. So what happens is the pupil does stabilize to an average baseline pupil size before you evoke any changes in it. But it is pretty dilated just because it's, the room is dark that the monkey sits in and the background's completely dark. So I think it would be super interesting to sort of do a parametric study and see if the evoked dilations scale sort of linearly or not, or in some really complicated way. But the other reason that your question is really interesting is because it'll also get at a little bit of what I think Doug Munoz is kind of getting at, either directly or indirectly, which is that it's the sort of balance between these two systems that might be really important to the dynamics of the pupil, right? And that's something that's never been studied, even in humans, as far as I know. So it would be, in principle, a fairly straightforward psychophysics experiment to do is to look at, set the baseline pupil size and then just evoke a dilation with some kind of startle stimulus, but for different, after giving the pupil different dynamic range to operate in and just see what happens. But I think with the superior colliculus circuit that Doug talks about, and the eating the Westfall, which are all heavily affected by luminance changes, I think that's part of setting the dynamic range of the pupil has got to be happening through that, and then how that balances with sympathetic tone. And we have no idea what that balance is or how it's perturbed. Like even in these measurements, we only began thinking about it after making all those measurements and said, well, these are one operating regime possibly that we've looked at, which is still interesting, but there's all the other measurements to be made.
You could also imagine motivational state having a kind of big impact over these kinds of circuits too. So like if an animal's really hungry versus if it's satiated, you can imagine this circuit kind of like the different kinds of things are salient to me when I'm hungry versus when I've just eaten. Like I could walk through the kitchen and if there's something cooking, I'm like, oh, wow, I'm super hungry. But if I've just eaten, I won't even notice the subtlety of the smell. And I think in part, a lot of these circuits, because they're so sort of baked in sort of in terms of phylogeny, that they kind of really, they're helping the animal find out what's relevant and focus their system's kind of attention, for lack of a better word, on that set of stimuli and not something else. And so I think, you know, it's obviously incredibly difficult to study that in the kinds of kind of empirical setups that you guys have, because you really want to have like really nice control and, you know, a nice, you know, relaxed animal, you know, it'll sit there and let you record for ages. But you'd really, you know, to find the breadth of all the different things that this system's doing, I think there's going to be a lot of really interesting stuff, I think, in that kind of, in that state space to explore.
I think Vinny Costa he's at Oregon Health Sciences. He has, I don't think he's done pupilometry, but he's been sort of getting at the behavior end of these things, sort of motivation, and he's recording in, I think, in BMPFC. I don't know if you know of his work, but that might be some really interesting papers to look at.
No, I'll check it out. Thanks.
Vinny Costa.
Sid, can I ask one thing on just in relation to you know what Matt pointed out, this the kind of spontaneous dilations that you see, which I believe are called the hippus of the pupil, and what I was sort of thinking of, though I don't know if you've actually tried to differentiate them, but I understand from when people got really interested in those fluctuations, like, you know, sort of a few decades ago, and they talked about them as slow oscillations, you know, and I think they find them in birds and mice and all species and stuff. I guess the way I sort of understood those, at least from older literature, is that they're always going to be a kind of, you know, relatively sort of slow kind of fluctuation in pupil size. So I guess I wondered whether would you always be able to kind of reliably differentiate them from a kind of evoked phasic pupil response? Because you know, I guess if you looked at the temporal derivative or something, these spontaneous, the hippus kind of things should always be happening at a sort of slower rate compared to the evoked changes, even if they reach the same magnitude, if that makes sense.
Yeah, great question. So two things that I've noticed is, one is they are smaller in amplitude than the evoked dilations.
Yeah, okay.
So that's one way of differentiating them. And the other is that they are on a slightly slower time scale. So the evoked, I actually didn't put that figure up here, but it's in the 2016 paper where we look at the startled sound evoked pupil dilations. Yeah. And so those tend to have, I mean, of course, those don't oscillate, it's just a single dilation and then the pupil settles down again. But those are on a time scale that's faster than hippus. If that answers partly at least.
Yeah, thank you.
Okay, so with this kind of microstimulation that I just showed you, if you take that data and then plot, sort of average it across all sessions, and in this case, it's 2 monkeys data, what we found was that the pupil size, when it's aligned to the time of microstim onset, shows this really nice stimulation link dilation. It's rather slow, but that's the pupil response. It's not like LC, or inferior colliculus, that have really sharp responses within 100 milliseconds. This is just the way pupil tends to dilate. We have seen individual differences as well between the monkeys. Like some of them tend to peak around 700-ish milliseconds, some nearly 900, which is quite a big difference in my view, as you know, used to thinking of time scales at the level of neurons. But this was one of the pieces of evidence for at least a part causal link between LC activity and pupil size. And again, you know, as going through the reviewing process, we had many discussions for this particular study, and it's hard to at least in our hands, we can't say whether this is a direct effect or not. It could be LC activating all of the other pupil machinery downstream, or part of it, or even some sort of loop via cortex. Who knows? It could be through ACC and then, back to some of the pupil circuits. But there is at least a link between activating LC and getting a dilation, that's for sure. So this is with stimulation, and then with recording, we kind of corroborated these measurements in different ways. So this is, again, from the 2016 paper. I'm not showing all of the areas that Mac mentioned. I'm just focusing on LC for now, but I'm happy to visit any of the others if anyone's interested in a specific one. So this plot is essentially it's the correlation between the mean pupil size in a single trial and the LC firing rate in that trial, and then calculated across trials. So the main result is that there is, on average, across all the cells recorded in all sessions, there's a small but reliable positive correlation between these two quantities, between the mean spike count and the mean pupil size, the mean spike rate and pupil size. That's measured in each trial. And the two data sets are from each from one monkey. So in each monkey, we see this reliable correlation between pupil size and LC firing. And this, we showed all of the data just to make the point that although, what we see is this positive correlation as a whole, there's a good number of examples where the correlations are neither positive or positive and not reliable. So there's a wide range of relationship between LC firing and pupil size. It's just that on average, it tends to be positive. So those measurements are for these average quantities measured, in a trial, but they don't tell us about direction because it's quantifying pupil size or LC firing in the same epoch, right? So you can't tell whether one happened before or after the other. And so one way of testing that is to do a simple STA, so this is spike-triggered average of the pupil size aligned to the time of a single LC spike. And so this is done for every spike from every neuron, and there's an STA for one neuron, and then it's averaged across all neurons and all sessions, and that results in this kind of plot. And so what we found was that on average, there's a dilation that peaks at about 300 milliseconds following single LC spike. So this gave us a little more confidence that there's some directionality in this effect, that it's a dilation following LC spikes. Again, tough to say if it's causal or not, because, again, it could be LC triggering something else, which drives the dilation. But there is a positive link between LC firing and pupil size. And another way to visualize this is to look at it backwards. So this is now getting to the point that both Mac and Claire, that you guys raised about hippus. So in these trials where the monkey is just doing boring fixation for long periods of time, we can find epochs like this that seem like one full kind of sinusoidal cycle of the pupil. And that allows us to find points of inflection, points of maximum rate of dilation and rate of constriction. And so we call these events. So a dilation event is just the peak rate of dilation in one cycle. And a constriction event is the peak rate of constriction in that same cycle. And then these red asterisks denote just the peak dilation and peak constriction. So these sort of points, what we can do is take the pupil and measure that in windows around these events, and then average that across all events. So that's what's plotted here. And if you look at the dilation and constriction separately, which we've done, and it's shown again in the paper, we actually see increases in LC firing rate preceding dilations and reductions in LC firing rate preceding constrictions in most cases. So we decided that the way to just combine that is plot the difference. So if it in fact is consistent, then what you're going to see is this peak in LC firing rate. And we found it's at about 300, again, about 300-ish milliseconds preceding these events. Okay, so now I'll move on to the newer data set, which is with the dual error recording. And
Sid, one question.
Sure.
With the ephys, with the LC activity, do you see it sort of oscillate like in time with the hippus oscillation?
Yes, sorry. Yes, we looked for it, and no, we don't.
No, we don't. Okay, fascinating.
No,
and then the other question.
Sorry, to finish your thought. At least not at the left, to be conservative as all electrophysiologists are, I think. Not at the level of how I was doing it, which is recording one neuron at a time in one track. There might be something if you're able to record, population of neurons in LC, because it could well be that as a population, it does oscillate with the pupil, but I don't have an answer to that yet.
Yeah, fascinating.
And I think no one's really recorded populations in LC, at least in awake animals, which is where it matters.
Yeah. Oh, that's good. I love how tentative all electrophysiologists are. Like, you're like, oh, it could be a directionality here. Like, it's like my old professors is very similar as well. Whereas we'd be like, oh no, complete evidence here, directionality. But my second question was, with how people always talk about in the locus coeruleus, tonic and phasic spiking, tonic and phasic locus coeruleus activity. Do you see that in like the ephys? Do you actually see like a baseline burst followed by like a really large change in firing rate?
So kind of yes and no, I guess. So in the sense that I really haven't seen spontaneous phasic events, honestly, in the conditions that we have, at least. So with monkeys doing boring fixation for long periods of time, which is up to 5 seconds, which is staring at a dot. Which is maybe not surprising because they're not doing a lot of exploring, or being offered different kind of stimuli to think about, or some kind of drastic change in the environment that might drive a phasic response. We do see evoked phasic responses. That's something that we showed in the 2016 paper, and I'm also going to show you more of right now. And so these are the kind of, we've shifted a little bit to calling it transient, and there's a long story behind that, because phasic in the old literature is kind of denotes a mode of firing rather than the form of the response. And so we kind of got into that in some reviews, including the paper I'm working on right now with this data. So we felt it's a little bit less controversial to just say transient versus ongoing. So when I talk about evoked responses, those are going to be transient evoked responses. The tricky thing with modes is that for that, you need to be monitoring a behavior over relatively long periods of time with changes in the environment. And we just started kind of getting to that with change point tasks in monkeys. But I think and again, the reason for this is for not getting to it sooner is we really feel like we need to characterize these baseline things sensibly first before making the measurements with complex behaviors. I think one could do it in either order. I just felt that getting the sense of this is better first.
Yeah,
so yeah, we'll be seeing some. Sorry.
Yeah, and so what like paradigms would you typically see like the clear transient effects? Like, is it in anesthetized animals, or is it in just like awake exploring animals that you would see the large phasic bursts?
Do you mean like spontaneous or evoked?
Yeah.
As in stimulus? Sorry?
In spontaneous.
So that's something we haven't done, but from the literature, it's in the anesthetized animals. So this is work that Gary Aston Jones's group did many years ago. I think the papers from the 90s that show in anesthetized rats, when you do, if you deliver a noxious stimulus, which is just essentially pinching the tail, you get this phasic burst in LC. And then I think there's work from, I can't remember, I think it's in mice with kind of a foraging task showing that you get epochs of phasic activity when the animal's exploring. But in our hands, we haven't quite done that yet, so I don't know but I expect it's going to be there, like and it would be really fun to do. So have the animal do some kind of foraging task.
Yeah, that would be amazing.
Yeah.
All right, thanks.
Ben Hayden, just to complete that thought, Ben Hayden in Minnesota might be, he started doing freely behaving monkey ephas. He's mainly recording, I think, in cingulate. So there might be interesting stuff coming out. I've seen preprints from them. They might, although I don't know if they're going to record an LC or not. That would be super interesting if they did. But
yeah, completely.
One setting. So one of the things we know about LC, or has been known for a really long time, is that it has really low firing rates. And in some sense, that's probably a good thing, because if it had high firing rates, we'd be anxious all the time, or hyper excited and hypervigilant. So this is a distribution of LC firing rates across all trials from all monkeys in the study. And you can see on about 30% of trials, there's essentially no spiking in LC during stable fixation. And on the remaining 70-ish% of trials, the firing rate is about a little less than 2 spikes per second. So this is interesting. Well, it's been well known for a while, so it's not super new. But what it lets us do is base an analysis of ACC activity on whether LC fires or doesn't fire. So if you take all of these trials where LC did not respond and treat them differently from the ones where LC did respond, and then look at what's going on in ACC at that time, then that sort of gives us a window into how LC might be having an effect on cortex. So the first thing I looked at was single neuron spiking in ACC. And what I found there is that there's no consistent measurable effect on single neuron spiking. So what I plotted here is the difference in ACC single neuron spike counts rather, for trials in which LC was spiking versus trials in which it was silent during stable fixation. And this is for each of the monkeys plotted separately. So you can see there's effects that are kind of noisy in both directions, not reliable at all. And I found something similar for the individual variability of ACC neurons. So this is quantified using fun factor. And here it looked like for this was interesting because with the first monkey, when I got the data set and I analyzed it, I got a bit excited thinking that there is a reduction in variability in cortex with LC spiking. And of course, monkey 2 came along and said, nope, not gonna happen. So that's what we have right now. So I can't really make any strong conclusion or any conclusion except that it could be either, but with an N of 2 monkeys, that's the story. But if you look at coordinated activity in ACC, so this is measured using pairwise spike count correlations for now. So this is, the measure is basically looking at spike counts of pairs of ACC neurons and seeing if they covary across trials in a given session. So that's the pairwise spike count correlation that we call RSC, R spike count. And I've plotted the difference for each pair, the difference in RSC for trials in which LC was active versus silent. And so the thing that really stands out here, I mean, it's a small but reliable effect, is that there is a reduction in ACC correlations, pairwise correlations on trials in LC is spiking, and that's reliable in both monkeys, whether you compare them individually, which is indicated by the filled bars versus empty open bars, I guess, or if you pool the data and look at all of it together. So for counting windows larger than about half a second, this effect becomes pretty apparent. And that's also interesting in a way because that argues for these effects possibly being mediated by LC, right? So again, this isn't causal because we're not perturbing LC, we're just recording passively from it. So these are associations between activity in one area and activity in the other. But the fact that, they're measurable only when we count in windows that are larger than a certain size, to me is a sign that it could be a modulatory effect, which is tends to be on longer time scales. So that's data from the trials in which the monkey is fixating and there's no external event. So in the same sessions, we also had interleaved trials where we played this loud kind of startling sound burst. And so this is an example of an LC neuron's characteristic response. Again, something that's been known since early days when Susan Sarah and Gary Aston-Jones reported things like this, that you get this, this kind of transient or phasic response to a startling event. So these dots, each line of dots is one trial, and each dot represents a spike. So this is a raster, essentially a spike raster. And then I've superimposed the PSTH on top of that. So that's just the average firing rate in gray, right? And so the characteristic response, as people have described previously, is that you have this sort of ongoing spontaneous firing rate of about 1 or 2 spikes per second, and then an evoked response, which for LCs, this is a huge amount of activity. It's about 7 spikes per second. And following the sound, it's suppressed below the baseline and then comes back to spontaneous. So that's, when you read the word sort of LC characteristic response, that's typically what it looks like. And that was 1 neuron. So this is the average of over 300 neurons from 3 monkeys. And it looks pretty similar. So there's this phasic activation and then a suppression below baseline and then a return to baseline. Just for completeness, I also plotted the ACC neurons responses to the sound. And this looks super interesting at first glance because it seems like it's an interesting kind of response, a biphasic spiking response to the surprising event. But in fact, if you look at the individual neurons, and I have that in the plot, I totally forgot to put it up here, but if you just make a color plot of the PSTHs of all neurons, this average tends to come from some neurons that have a phasic response and some that have a suppression. So we don't find many single examples that have this profile. So this is more like a population profile. So it's a little bit misleading if you're thinking in terms of single neurons, but it could be interesting if you're thinking in terms of populations. So with the sound, there's also a change in the individual variability. So on the left, I've plotted the fan factor for locus coeruleus neurons aligned to the time of the sound. And you can see there's this sort of sharp dip in the fan factor. And we're not, in case any of you were worried, we're not violating causality here. It's just you have to use big windows to count enough spikes in LC to get a measure of the variance. Otherwise, LC is barely spiking. So if you make the windows small enough to get the exact time, it gets very noisy. And ACC is similar, although it looks like maybe a more sustained suppression, reduction of individual variability following the sound. So then we ask whether the sound also has some effect on the correlative activity in ACC. And if you look at the entire population of pairs from the ACC recordings, there's no single sort of simple reliable effect in any direction. So there's increases, the decreases in correlation. And so it's, it's not obvious. There's no obvious effect, I should say. But if you look at what LC is doing on those same trials, and something really interesting does pop out. So although me and others use the term characteristic response of LC, if you look at trial by trial responses, you see something different from what you might expect. Certainly different from what I expected. I thought there'd be a reliable phasic response to every startle stimulus. But the point is, we're not monitoring the animal's internal state in a very reliable way, right? So on trials where we think the stimulus is going to be surprising, it might not be. Like LC might already be active at a high baseline level. The animal might be in a higher state of arousal. Whatever reason, like someone walks by in the hallway, they're very sensitive. They can hear things, hear what's happening outside their booth. So essentially what I found was that in this plot, this is the proportion of LC neurons that have that many trials in which the phasic response was reliably evoked. So that's pretty, I mean, to me, that's really striking. It means that about, on average, what we call the characteristic LC response is evoked on roughly half the trials in a given session, on average. And on the remaining trials, it's either no response or really weak response compared to baseline. And so this allowed us another sort of tool to get at what might be happening in ACC. So it's not enough to just play the startling sound or have a sudden change in the environment. You also have to know what's happening internally. And so on trials in which LC had a phasic response, what I found was that the correlation, the pairwise correlations in ACC actually increased pretty reliably for all sizes of counting window that we use, compared with trials in which LC did not have a phasic response. So those are the black circles. And again, these are filled or open depending on whether the effect was reliable before, after relative to before the event. And then these markers indicate whether the phasic and no phasic groups are reliably different from each other or not, per pair. And then to test whether spontaneous phasic-like events in non-startle stimulus trials might also drive these kind of changes, we took the trials that did not have any sounds and then generated a random time that we called a beat time and aligned spikes to that, and then looked for spontaneous events that are similar to phasic evoked events from an actual sound. And then did the same calculation again for ACC pairwise correlations and found that there's basically nothing, which is interesting and reassuring, because what it means is that this effect is an external sound-driven change in LC firing that is either affecting, I don't want to use that term again, but it's consistent with, and it happens on the same trials in which LC responds compared to when it doesn't respond. And so kind of actually I was going to go to an interim summary, but I guess I had another slide in there. If you factor in pupil, and this is where it does get interesting, then this effect is a little bit larger. So if you look at the y-axis on these two plots, if I do the same calculation, but now I factor in whether the sound also evoked a pupil dilation compared with when it didn't, then the effect is still there. In fact, the magnitude is a little bit larger, about 0.01. And if we factor in the baseline pupil, which is also interesting because we expected there to be sort of a relationship between the two, it does not in fact seem to have an impact on the measurements in ACC. So it's really the evoked pupil that matters, not the baseline in this case. So quick interim summary is just to drive home the point about the directionality of this effect, because that to me was the most interesting in the study, was that when there are changes in ongoing spontaneous activity in LC, or rather when it's active compared to when it's not active at all, then we see a decorrelation in anterior cingulate. And when there's an event-driven increase in LC firing, then we see an increase in correlated activity in anterior cingulate. So these are, I think it's really interesting because it suggests that, especially if you look at previous work, both with the cholinergic system and norepinephrine, there's this trend to, think mainly in terms of decorrelation of cortical regions. And a lot of that work's been done in sensory areas. I wish we had sensory recordings here to compare this with, not just ACC, but we don't right now. But I think what we're seeing is similar to what people have shown for in sensory areas, is that you do get decorrelation and all the other things that follow from that, better signal to noise ratios, better information encoding, et cetera, that people, argue about. But in addition, the same system might be driving, synchrony at some time scales when it's an evoked response in that system. So it's that, it could be a switch that switches the cortical state between decorrelated and correlated, depending on whether LC was activated in one way or the other. Okay.
So just at that point there, Sid, is actually kind of an interesting point of sort of overlap with some stuff that we've done in the past, which is to think a little bit about what noradrenaline might be doing to kind of populations of neurons, and in terms of changing their receptivity to one another, and their sort of ongoing activity. And there's a lot of assumptions you have to make to do this kind of modeling, right? You have to kind of make a lot of, you have to pretend that the mean population is a pretty decent, mean population activity is like a decent kind of summary measure of the activity, and you forget a lot about the idiosyncrasies. But when you do that, you can, use, we've got a couple of papers that have looked at this. You can sort of think of noradrenaline sort of changing the receptivity of the neurons to one another. And one kind of really cool thing that happens, just to kind of give you, Joe, a bit of an olive branch here to kind of link to, is that some of the measurements that you get from these kinds of models, you can then put them through the kinds of analytic tools that we use. Like Joe's really interested in thinking about information sort of in terms of the reduction of uncertainty in a signal, both within an area, but also between areas. So in the first case, it would be how much, if I know the past of an individual neuron or sort of region, how much can I tell about its future? And then the second one would be, if I know about the past of neuron A, how much can I predict the future of neuron B, or reduce my uncertainty about neuron B's time series? And one of the things we find when we manipulate a variable that we would sort of, that relates to this receptivity that we think is a sort of noradrenergic mechanism, you see this big switch between those two modes where in the sort of low noradrenaline state, in inverted commas, you see a lot of self-storage. But as you increase this variable of this sort of gain change, what you effectively find is this really abrupt change, which physicists, you have a physics background, we think of it as a sort of a bifurcation, really abrupt change that looks similar to a lot of critical processes. Now you see this big shift towards much more information transfer and a much more influence between regions. So it's really neat that you're finding similar kinds of effects empirically, because from our perspective, through a ton of assumptions, really simple neural models, we're kind of finding similar types of patterns. And we're really keen to kind of embrace this idea. But I just thought I'd throw a little olive branch to you, Joe, just so you had some tether to this world of neurophysiology.
Thank you. Definitely appreciate it. I was thinking similar things.
Could I just ask, if possible, would you mind just going over how you measure the correlations in the ACC again? That's a critical feature that I seem to have managed to not understood, so apologies for that.
Oh, no worries. So these are spike count correlations between pairs of ACC neurons. So all I'm doing is, it's based on a measure that I think it's been used for quite a bit. It was, the paper I'm thinking of is Baer and Zohari and Newsom from 90, I'd like to say 6 maybe, or earlier, thereabouts, where they're measuring correlated activity in MT, area MT, that's the motion selective area in macaques. So it's basically for every trial, we measure the spike counts of an ACC neuron, and then do the same thing for every other ACC neuron recorded simultaneously, and then take them in pairs and measure the correlation between spike counts across trials for that pair. When we do it in a binned way, it's doing the same thing now, but then just aligning all the bins. So it's taking or I should say window rather than bin. So if it's for a 500 millisecond window, you start at the start of fixation, of stable fixation, and move that window with some step, with some shift, and count spikes. So there is some overlap in the windows. We've done these sort of multiple comparison tests to check that it's not something that's coming from random correlations.
Right.
I'm happy to explain if that didn't make sense.
So, and I'm not sure why. No, that's fine. That makes sense. I'm just wondering. I don't know why I'm wondering, but are these measured from the same electrode? These pairs of neurons, are they from different electrodes, or?
They're from mostly from different electrodes. So I didn't mention, for the ACC recordings, I use a linear array that has 8 to 16 channels, so 8 to 16 distinct sites. And so after recording, we typically do something called spike sorting, which allows us to use these clustering methods to make sure that we're not assigning spikes from two different neurons to the same neuron.
Are there regional variations within ACC then, or you haven't got enough data to look at that?
You mean from different tracks and different bits of ACC?
Yeah, I mean, I don't know what this array, how much it spans, and there's, I mean, I'm way out of my comfort zone asking these questions too, but it's curious. It's just
they're great questions for an electrophysiologist. They're fantastic questions. So I think the short answer. Let me just go back to this not so great schematic from Paxinos. It's the standard monkey atlas. I just picked this slide from there. So this is generally the region we're aiming for. You can see my mouse, right? The cursor?
Yeah.
Yes, okay. So 24C is, well, actually all of this is considered dorsal ACC. Ventral is down here. So these are areas 32 and 24C. And the way we figure out where we are, because we don't have, at least in Penn, we don't have this sort of very high resolution MRI that, you know, places like Riken have, where they can image the electrode and the layers. But one way of mapping or figuring out where we are and sort of, I think, getting at what you're asking is to do single electrode tracks to first map all of this area, because it's otherwise difficult to figure out where you are in ACC. It starts getting a bit cramped at this very frontal location. So what we do is we typically start in the middle of the recording chamber, which lands us somewhere here. And then you kind of look for gray matter, white matter transitions, which you can tell just by listening or looking, because in the gray matter, you see these nice sort of big extracellular action potentials. And once you get on the white matter, those disappear.
Right.
So you can, you know, if you move about 0.5 millimeter at a time in single tracks, which is what we do before starting the main recordings, then that you can sort of step your way towards the more medial side. And also technically, we don't start very much medial because they're typically large blood vessels running along the midline. And you definitely don't want to have to go through those.
Yeah.
So if you sort of tiptoe your way through from the lateral edge, that's safer. And you also see these transitions. So once you get to this gray matter, lots of white matter, and then gray matter, white matter, back to gray matter, then you can be pretty certain that it is this bump.
Right.
And the orientation is relatively normal to the surface. So I think what we're doing is going through, the electrode's probably spanning about 2/3, given it's the length of the region where the sites are located. It spans the pale surface to white matter width of 24C. And we didn't try very many locations. Once we find a good spot, it's maybe 2 or 3 locations around that. That's a long answer, and it doesn't answer your question.
No, it doesn't. That's very interesting. It's helpful to go back to the anatomy.
Right. I haven't yet looked at whether those physical locations get us different results. That is a great point to bring up. I think I might dive into it at some point.
Okay, great. Thank you very much. Thanks for explaining that.
And Sid, while we have the anatomy up quickly,
Yeah.
so there's reciprocal connections between the ACC and LC.
Mm-hmm.
Yep. Okay. So they both drive each other, or
In the set, yeah, that's so the LC projections to cortex are very diffuse and they're not driving connections. So that's one thing to be a little bit careful about when you think about I think any of the neuromodulatory projections, is that they are not directly synaptic. They diffuse and they sort of, you know, kind of like a fire hose, essentially. Although at a much lower flow rate, they release norepinephrine in the neuropil, and then whatever receptors happen to be nearby on the dendrites of cortical neurons, those bind norepinephrine and then open up whatever channels they do. And so that's getting to a level we can't get at with extracellular recording. But just to say that they're not driving input. So when you see these effects of LC activation, again, either effects or, you know, covariant phenomena, then it's going to be on longer time scales. I'd be surprised if it was shorter. But then the reverse direction is synaptic. So the projections from cortical regions and some other subcortical bits to places like LC and Raphe and nucleus basalis, those are synaptic. So those are driving inputs. And that's the bit in the motivation slide I mentioned, I think I'm super interested in, is figuring out, if we find a spot, again, it'll be hard to do. I'm not sure exactly how to do it, but where there's a patch of neurons that we know they have projections to LC, and see what those ACC neurons are encoding. Are they the ones encoding cognitive surprise or not? Because we could record from hundreds and thousands of ACC neurons. They might be all the ones that are not projecting to LC. So to understand what's driving LC under these higher cognitive conditions, there is a way of doing it actually, other than the super fancy new methods, is to do what's called a collision test. So you stick an electrode in LC and stimulate. So you pass current, and then you look for rebound excitation in the projecting neuron in ACC. But that also means you have to hunt for that neuron, because they could be 1 in 1,000 dispersed throughout ACC. But it could be worth it.
Yeah, that's fascinating. And then I guess the other one, this is also amazing work as well. I love it. Like, it's great to hear any phys person speaking at this. It's I don't know why. It's very, I love just like the detail that all these experiments are done in. But what do we think? So why does the ACC typically have like these like kind of, surprise responses? Like, is that like a known thing that the ACC sort of like, has like a response to surprisal events?
In the data I showed, the things I was hand waving about from other people's work.
Yeah.
So I think the kind of, we see these responses to the startling stimulus in some of the neurons. And I think that's related to things like cognitive surprise or violation of expectation, violation of priors, which is what people have measured. So I think I can't remember the exact authors, but Michael Platt's lab has some ACC recording papers where they show these I forget if they showed cognitive surprise or performance monitoring kind of signals. I'll have to dig the papers out. I can't remember the exact authors right now. But using different techniques, including ERPs and MRI and EFIRs in mice and monkeys, people have shown that there are these signals that are related to surprise, and things that it's got to be cortex that controls those, because you don't see those kind of signals in thalamus or in other brainstem structures. These are quantities that we know, and others have characterized as being computed in areas like medial prefrontal and orbital frontal and ACC, in fact.
Mm-hmm.
So if a simple task would be like even playing a series of sounds and then having kind of like an oddball and then building up a prior for what that sequence of sounds is, and then having an oddball in that, right? And the fact that that's an oddball is not encoded further downstairs. It's all happening somewhere in cortex. And ACC is one of the regions that seems to encode those.
Yeah, okay, thanks.
Just to link it to something interesting, Brandon, in your recent paper, looking at that Hassenkamp data, when they did a bowl analysis of the blood flow, like the bowl with the GLM in the traditional analysis, they found that the anterior cingulate was active just before the button press, so right around when we saw the LC peaking as well. So there's kind of really interesting kind of evidence, and a bunch of people have theories around this at that kind of systems level about anterior cingulate and in the anterior insula being part of what they call the salience network. Is these sort of broadly distributed areas bilaterally that respond to salient events, either internally or externally salient. So it could be like your heart starts racing and then you notice it, or someone sort of shouts in the room, and either of those kind of get funneled through this system that then the sort of canonical idea was that it kind of shuts down your endogenous attention. You stop, mind wandering or, focusing on some particular, part of, something that's, internal, and you orient your attention towards the external world such that you're kind of like ready to take in the new information. That's the kind of, it's like a Vinod Madone and Lucina Unen had a really nice kind of framework for this back in the 2010, 2011 kind of space. But it's cool. So it's really cool to see this down in like the electrophysiology as well, away from the kind of blood flow space where things can get a bit murky.
Yeah, block your ears, Rob. I just don't know enough about the cingulate.
Matt, can I ask you can I ask a question that builds on that? So in the when the salience network's activated, does the cingulate sort of coordinate that sort of broader activation? And could there be the reciprocal connection between the cingulate and the LC be important also for that? And do we know?
I mean, yeah, maybe Sid has a better answer than I do, but from my understanding, the like biological mechanism of this is very poorly understood. There's lots of empirical recordings. You can go back to like the mid 2000s when Fox and colleagues, Reichel and so showed this kind of anti-correlation between these kind of more endogenous areas like the default mode network and the kind of more exogenous attention, like the dorsal attention network regions. And then the next story from that was that something like the cingulate was controlling whether or not you were in either of those modes or the insula, but whether or not you could like go down to brass tacks and say, okay, a pyramidal neuron from anterior cingulate is going down and actually shutting this off and shutting this, turning this one on. That level of explanation is still kind of, an open question. And there are folks like, I know Helen Barbas's lab at Boston are thinking about this stuff and trying to work out, which Brodmann area could be projecting to which other one, and which way that could cause the system to shut down or increase its activity. And it's kind of complicated when you think about it. And there's probably going to be a big role for subcortical structures like the habenula and parts of the thalamus, the zona inserta. There's all these structures in the subcortical kind of space that have this really intricate interactions with one another that are excitatory, inhibitory, and balances between them that I think really need to play a role there to kind of make sense of it all. So I don't know if Sid wants to add anything. It's not super my space.
Largely agree. To directly answer that question, you kind of have to, at least with EFES, you've got to do some kind of population recordings in pairs of regions or triplets of regions. And that's just not happening yet. Even with LC and any other area, this, as far as I know, this is pretty much it for dual recording. And I'm actually surprised because the techniques for doing this are out there and they're very efficient in the sense that you don't have to do the painful, single electrode or small numbers of neurons, which was the case even 10 years back. So now you can do up to 128 or 256 channel recording with really fine microprobes in a couple of regions in cortex, in pretty much any model. So yeah, I'm a little bit surprised that there's not a lot happening, or maybe there is, and it's in the process of coming out now. You'll see it soon. But as of now, yes, I agree. We just don't know whether these sort of theories pan out in the way people are thinking. I suspect they won't be very far from the truth because they roughly make sense. They're consistent with what's happening in these different regions. I think the details aren't there yet. And one bit that I'm really interested in that's related to this ACC, or indeed any prefrontal region that has projections to LC, but and knowing that sensory regions don't, is that in perceptual behavioral tasks, that loop would be really interesting to explore, right? Because what it suggests is that the fidelity of ongoing information coding in sensory areas can be modulated in real time by LC, and it can switch sensory regions in the way that found here for ACC. It can. Now, we don't know whether the same effect that I've seen in ACC also is generalizable to other cortical areas or not. I hope it is, because then it gives us sort of a more unified picture of what LC might be doing, and later maybe explore other modulatory systems as well, like serotonin. But that would be super interesting to find out. And it would take, technically just recording in LC and V1, for example. It's not that hard an experiment. But it's tough to get to do it, I think.
Yeah, I want to see a monkey just with like 10 neuropixels loaded across the brain. I feel like we're going to get some answers that way.
That might be too much data. there is such a thing. Okay, how are you guys doing for time? I have maybe a small number of slides. I'm happy to either go on or stop. There's a cutoff.
I've got a hard deadline at 11, but I'm enjoying the chat. If people need to go to other things, no hard feelings. We've got a recording. I'm happy for you to keep going if you've got time.
Sure. Let's see. Just reminding myself where I was. All right, so these were the LC condition measurements of ACC spiking. And the next thing I wanted to look at was these similar measurements, but now related to ongoing pupil fluctuations. So this gets back to what we were discussing a few minutes back about spontaneous pupil oscillations during fixation. So if you take the trials when, again, these are fixation trials without any external stimulus, the pupil tends to have this fluctuation that's also called hippus. And so we can identify points of peak rate of dilation, peak rate of constriction, the peak dilation and the peak constriction. And so one way to quantify this is to just assign them phases. I mean, you can do it any way you like, but one way is call the peak rate of dilation 0 in a given cycle. And then the other points are assigned these phases as shown here. And so you can again then analyze the LC firing rates and the correlated activity in cingulate cortex as a function of, or aligned rather, to these phases. And so when you do that, what we see is, so in the top plot, this is the PETH, or the peri-event time histogram of LC firing. The colors are basically firing rates indicated by the color bar here. And what we find is that there's this sort of systematic change in the firing rate with phase, almost like, I forget the word for it, it's like, what's the term that's also seen in hippocampus? Precession or something.
The precession, yeah.
And the neat thing was, in our previous paper, we looked at essentially this point. So there's a figure that shows the, I think it was in this talk as well for the back, where I plotted the pupil aligned spiking in LC. So if you look at only the peak dilations and look at and look for when the LC spiking peaks relative to the maximum rate of dilation, that's essentially a 0 phase. And we found, if you remember in the plot that I showed a little while back, that peaks at around 300 milliseconds before the event. And so if we, here I did essentially the same analysis, but now for all phases, or rather phases stepped in 15-degree intervals that I extrapolated from the pupil oscillation. And that's interesting to see, and also reassuring that the numbers match with what we measured in a different set of monkeys a few years back. And the correlated activity in ACC also shows this kind of precession that seems to be related to the timing of DLC firing. And so, just eyeballing these plots, it looks like there's an increase in LC firing at these delays relative to each phase. So for phase 0, the peak is here. For phase 90, the peak is somewhere here, and so forth. And then in ACC, if you just shift that curve a little bit, there's a reduction in relative reduction rather in pairwise correlation. So that's a little bit consistent with the measures I showed you about ACC being decorrelated when LC spikes. The other interesting feature that popped up, which we hadn't expected, was that there's actually an increase in correlations preceding the increase in LC firing. And that was kind of neat because it means that, again, this is probably a kind of a loop, right? Correlated firing in ACC goes up, preceding, on average, preceding increases in LC firing, which is what's happening, what seems to be happening here. And I have some more analysis for this coming up to quantify it a little better. So related to this, we want to just look at what the timing is of the changes in LC firing related to the timing of changes in ACC correlation. So remember the plot that I hate doing this, but switch back to this plot. So these are not measured, these are measured in windows relative to fixation start time, but these are all aligned windows. So these are not relative, it's not ACC relative to LC, it's just all the bins relative to start of fixation for LC and for ACC. But if you now take those bins separately and calculate both the firing rate in windows around the time of start of stable fixation, before and after fixation starts, and so this is LC firing rate, this is ACC pairwise correlations, right? Then some sort of interesting features pop up. And one of them is that this increase in LC firing rate that you see before stable fixation starts is essentially due to the saccadic fixation eye movement. So the fixation point comes on somewhere back here at minus 11 or 1200 milliseconds. And we can't align it to both in the same graph. So I've only aligned these to the time that the eye is stable within the fixation window. And so when the fixation point comes on, it's sort of a startled stimulus, and the monkey makes a saccade to look at it. So there could be two factors driving this. We haven't eked them out yet, but we know that from the 2014 paper of Rishi's, and others have shown this as well, that there's an effort-linked increase in LC firing across many species. So whenever there's a motor action, whether it's an eye movement or it's a mouse running on a wheel, there are increases in LC firing. So I think part of this increase in LC firing is from the saccade to make, that the monkey makes to get to the fixation point. And the other could be a little bit of a startle because each trial starts with a random inter-trial interval. So the monkey can't predict it exactly, although they probably get pretty good at memorizing the distribution after a few years. And then after stable fixation starts, not surprisingly, LC firing kind of levels off. There's nothing interesting happening. It's just tonic spontaneous activity of about 2 spikes per second, which we saw earlier. In those same epochs, the ACC correlation, so I plotted it here separately for trials in which LC is silent and trials in which LC is active. And one of the interesting things is that for trials in which LC is silent, correlations are larger. So again, this is consistent with our collapse window calculation, right? Where we've taken all the windows together and calculated 1 grand correlation coefficient. So what this shows is that actually over the entire course of the trial, in fact, starting before the fixation or just when the fixation point comes on, if LC is active, ACC is less correlated. So those are the heavier lines down here. And in one attempt this is not finalized, this is sort of very preliminary analysis in this last plot, I just took the cross correlation of the average PSTH and the average ACC correlation to see if there's, if that timing makes sense. And by that, I mean, it looks like there's this bump in ACC correlation early on when the fixation point comes on and the monkey makes a saccade, and then it declines. And then following that increase, which happens around 1 second before fixation starts, there's an increase in LC firing. So to me, this seems a lot like this increase leading to that increase, except they're measured in separate epochs. But it doesn't matter, because if you do a cross correlation, it should tell you the temporal relationship at different lags, right? We're still working on some of the details of how to quantify that. And I don't know if you're going to show exactly that in the paper or not, but it does seem to show. So this is the correlation for ACC on LC spiking. And so there's a bump, there's an increase in ACC correlations before increases in LC firing. If you do the same thing for trials in which there are evoked responses in LC, so again, these are divided into trials when the sound evokes a response. So this is the phasic or transient burst in LC. And these are trials in which there was no transient burst. And we look at ACC correlations in those sets of trials separately. in both cases, it looks like the sound drives an increase in ACC correlations that's relatively slow. So yeah, it peaks after 500 milliseconds. But the interesting bit is in the slope in this region. So when LC also fires, the slope is a lot sharper in the rate of increase of ACC correlations is sharper than when LC does not fire. And this difference is really reliable. It's surprisingly reliable. Initially, when I thought of these and I was just staring at the peaks, I thought there wasn't anything super interesting happening. Then I started looking at the slope, and there was, in fact. And these again are the cross correlations between these sets of curves for each epoch measured relative to the other. Let me just check that there's, okay. And finally, I'll probably stop at this slide. This is a measurement that's similar to these ones, kind of just a sanity check, is to take, rather than doing a moving window measurement, is just divide the post-beep epoch into the first 500 milliseconds, the next 500, right? So an early versus late response. And then look at whether ACC correlations in those two epochs are different for the two kinds of trials, when LC was active or not in response to the sound. So in the early phase, there's no difference between ACC, RSC. There is a difference in the slope, but there's no difference in the mean correlation. But later, there's a clear difference, and that's reliable, which again, suggests that the fact that there's a difference in the time course of release of norepinephrine is affecting how fast ACC correlations change, but they eventually get to about the same level. Okay, I think I could stop there.